Katie is from Loveland, Colorado, so I feel bonded. I also grew up in Colorado, not in Loveland. That's okay. I remember kind of the first experience I had with Katie was in atmospheric and environmental chemistry, kind of, and we made this beautiful poster about the timeline of air pollution, and it was the first time I really realized how well Katie could communicate and just kind of sort out ideas in wonderful ways. And since then, I really feel like I've been privileged to see her grow through analytical and then an instrumental analysis, where we really started to find this niche of wanting to look at kind of coding and methods and more large data, and how to look at some of the most interesting analytical aspects, whether it be biochemistry or more related to computer science, but really on the edges of interdisciplinary work. And I could not have been more excited for her to get her RU that she's going to talk about for this talk out at Stanford, because it allowed her to really find a place where the chemistry and the question really spoke to her. So I just am so proud of how you have grown and changed, and I feel really privileged to have been able to work with you throughout your career. So I hope that you enjoy this talk, because I think it's something a little bit different that you don't get to see here at Lawrence all the time, and just a wonderful opportunity. Yay.
At Stanford, developing a contrast agent for early breast cancer detection. And as a quick disclaimer, there's a lot of breasts in my presentation, but let's free the nip. So to give you a bit of context behind my interest in this area of research and how I was able to participate in it. So leading into my junior year of college, several family members of mine were dealing with cancer diagnoses. And so I was like, okay, well maybe the science I'm learning can help out with this. So I looked for programs that focused on cancer research, and I found one at Stanford called the Canary Crest Program, and they particularly focused on early detection diagnosis techniques as well as early stage therapeutics. So I applied during the winter term and believed, okay, I don't have much of a chance of getting this. I mean, it's at Stanford, right? So I went abroad to the London Center, and it wasn't until the end of my time there that I found out I actually got this position, and I was super excited. So I hopped on a plane and flew back to Colorado, where I'm from, and then drove out to California with one of my aunts who had a lot of experience with breast cancer because she had been diagnosed with it. So we had a long conversation about her experience. So while I was in California, I met a ton of amazing young women. And I also worked closely with these people all summer. This is my lab group. So on the left, this is Katie Wilson, and she was my primary mentor. And so she's an instructor in the radiology department at Stanford, and she focuses on the development of a novel imaging technique called photoacoustic molecular imaging. And then on the right-hand side here, this is Rakesh, and he was a graduate student that I worked with closely throughout the summer, and he loved yeast. And I had a third mentor named Lotfi. He's not pictured here because he moved away in the middle of the summer, but his primary focus was on protein engineering. So all of their strengths came together to put together this project. So as I was traveling out to California, I realized I don't know all that much about breast cancer, and I started to do some research. And I found out some stuff about breast cancer statistics. So breast cancer is the most common cancer among women, and so if you are a woman and you are going to develop cancer in your lifetime, it's most likely going to be breast cancer. So it's responsible for 30% of all new cancer cases. So that is around 268,600 new diagnoses in this year. And it's also the second leading cause of cancer-related mortality in women. So that's around 41,760 deaths in 2019. And that's right behind lung cancer. So next I looked into the biology of the disease, because despite having breasts, I wasn't really aware of what lies underneath the top layer of skin. And so I found out that breasts are known as mammary glands medically, and they're made up of lobules, which is a milk-producing structure, and that's this stuff here. It forms a circle around the nipple, and it's connected to the nipple through a system of ducts. You You also have a ton of lymphatic vessels which run through the breast, and these help drain excess fluid as well as bring in important immune cells to fight off inflammation and disease. But primarily the breast is composed of fatty tissue and connective tissue, and that's this yellow stuff here. And I also want to note that breast cancer isn't primarily affecting women, it also can affect men. And their anatomy is almost identical, except they lack the lobules and ducts seen here. So what are the stages of breast cancer development? So the early breast cancer stages are stages 0 through 2. So stage 0 is non-invasive carcinoma in situ. And I'll go into what carcinoma in situ means in just a bit. Stage 1 is you have developed a tumor that's just under 2 centimeters in diameter, and there's no evidence of metastasis. Stage 2, the tumor is still localized within the breast and only extended to a nearby lymph node. The later stages of cancer are stages 3 and 4. So in stage 3, cancer extends beyond your immediate lymph nodes, but not to distant organs. And in stage 4, cancer is spread to distant organs. In other words, it has metastasized. And in the case of breast cancer, the first place that it will likely metastasize to is your lungs. So for these 5 stages of breast cancer, what is the 5-year relative survival rate following diagnosis? So if you get diagnosed with breast cancer in its latest stage, stage 4, your chances of survival are around 27%. It's not very good. However, if you can catch this disease in its earlier stages, stages 0 through 1, your chances of survival are around 99%. And that's kind of the premise behind the Canary Crest program, is in funding research that focuses on diagnosing these cancers in their early stages. So what is stage 0 breast cancer? And this is primarily focused on women since men don't have these structures. So stage 0 can be divided into 2 types, DCIS and LCIS. DCIS stands for ductal carcinoma in situ, and that just means it's developed in the duct and remains there. Lobular carcinoma in situ means that it's developed in your lobes and is still localized there. So how do physicians currently go about screening for breast cancer? Current diagnosis techniques include mammography and ultrasound. And occasionally MRIs may be used, but my primary focus is on these 2 techniques. So despite knowing mammography was something I had to look forward to in my future, I had no idea what it entailed. And I also asked several peers of mine, and many of them didn't know either, so I found a video to demonstrate this. So this technique works by placing the breast on a support plate and compressing it with another plate. This is necessary to reduce path length and stabilize the breast, so then when you shine low-dose x-rays through the breast, and it'll meet the detector down here, that way you get a clear image, which looks something like this, a mammogram. So on a mammogram, fat will appear translucent or this lighter gray color. And denser tissues, like your tumor would be, will appear this whiter, and this is like connective tissue. But this image shows one of the biggest limitations of mammography, and that's breast density. And unfortunately, breast density afflicts 50% of the women who are likely to develop breast cancer. And also I want to note that mammography starts when a woman's around 40 years of age, and it's a yearly screening until you're around 55 years of age, afterwards throughout every other year. So it's a big part of a woman's life. So if a physician suspects breast cancer, that you have breast cancer, or you have dense breasts, they'll ask you to get a diagnostic ultrasound, and this is a relatively harmless technique. So a physician will place jelly on your skin and then use a transducer to emit sound waves into the body, where it'll hit tissue boundaries and bounce back, providing an image that looks something like this. And this is a 2D image, which provides pretty good tumor visualization of big tumors. But it's not going to be all that effective in the early stages of diagnosis. And also, a big limiting factor is that ultrasounds have a high rate of false positives. So this leads to unnecessary biopsies and callbacks. So breast density is a major risk factor for breast cancer development, and this afflicts 50% of women. So there is a need to improve upon current imaging techniques. And the lab that I went to at Stanford was focused on photoacoustic imaging and fluorescence imaging. So what is photoacoustic molecular imaging? And I found a quick video. Or not. Okay, well, I'll summarize it for you. So this technique uses light and sound waves to visualize tissues in the body. So near-infrared light in short 10 nanosecond pulses irradiates the tissue, and chromophores will expand and contract, releasing acoustic waves, which then can be detected by a transducer. And it produces an image that looks something like this. So you have a 2D image, your 2D ultrasound image, but you have better optical characterization of the tissues. So the other technique we focused on was fluorescence molecular imaging. And basically, a fluorophore has electrons at a stable ground state, but when we irradiate the fluorophore with a given UV wavelength, it gives the electron the energy to jump up to an excited state. There is then an internal conversion between the rotational and vibrational energy levels, and it is ultimately unstable in this state and will fall back down to the ground state, emitting a photon of lower frequency, higher wavelength in the process. And that produces a spectrum that looks something like this, with wavelength on the x-axis and light intensity on the y-axis. Absorption is at a lower wavelength, while your fluorescence emissions peak is going to be at a slightly higher wavelength. And this is the image that is produced. So, some important results of prior research done by this lab at Stanford included identifying a marker on breast cancer cells which can be targeted for imaging and therapeutics. This marker is called B7H3, and it's a receptor protein on the cellular membrane. It's specifically expressed on endothelial and epithelial cells. And endothelial cells, they line the interior surface of blood vessels and lymphatic vessels, while epithelial cells are a type of cell that lines the surfaces of your skin, blood vessels, urinary tract, and organs. So once they identified a marker on breast cancer cells differentially expressed on breast cancer cells, they decided to target it. And so they created an antibody ICG contrast agent to aid in tumor visualization using photoacoustics and fluorescence molecular imaging. And so for those of you who aren't aware, an antibody is an important component of your immune system, and it has the capacity to bind to targets with high affinity. And ICG is just one of the fluorophores. So to help visualize this, I came up with an analogy. And so if you think about Velcro, and you consider that this B7H3 marker is kind of like the base of Velcro. And so your cell is covered in it. And the antibody is more like the hooks. And so they bind to each other with high affinity. And these scientists, what they've done is glued a mirror on top of the hooks of the Velcro. And so that way, when we shine light at it, we can identify our cell. And so then when you're looking at a large pool of cells, normal and cancerous, you can actually find your cancer cell. So what are the disadvantages to this? Well, antibodies are expensive to produce, and they can cause an immune response in humans. So, our research goal then is to create a contrast agent for photoacoustic and fluorescence molecular imaging that is inexpensive to produce, won't induce a large immune response, and targets breast cancer cells with high specificity and sensitivity. And we considered doing this with an Affibody. So what is an Affibody? It's a 3 helical structure derived from the Z domain of Staphylococcus aureus protein A, and a yeast library may be used to engineer a binding ligand for a desired target. And this is a really cool technique because it allows us to engineer proteins which bind to targets with high affinity. And I don't know all that much about this because the University of Minnesota actually did this for us. So an Affibody is pretty small, it's around 7 kilodaltons. It has fast tumor infiltration, a low immune response, it's stable in a wide range of physiological pH and temperatures, and it's cost effective for large-scale production. So the next step to creating a contrast agent is deciding what fluorophore to use. and we decided to use indocyanine green, abbreviated ICG. And this is the molecule here. As you can see, it's highly conjugated. And so it's an FDA-approved near-infrared fluorescent dye, and it's pretty small, around 818 Daltons. And it absorbs primarily between 600 and 900 nanometers, and fluoresces then between 750 and 950 nanometers. So now we need to figure out how we're going to create this contrast agent. And this can be divided into two main synthesis steps: Affibody production and then Affibody dye conjugation. And then we need to characterize it using nanodrop UV-Vis, Sds Page, and MALDI-TOF. And I'll go into what these mean later. So first of all, Affibody production. This was pretty simple. It was done in 3 steps: cloning, expression, and purification. So in the cloning step, we need to choose an appropriate plasmid, and a plasmid is just a circular piece of bacterial DNA. Then we insert a construct, and a construct is a gene of interest, whatever you want the bacteria to produce for you. Then we transform it in E. coli, allow E. coli to grow it, and then we purify it. So we did this using FPLC, which is called fast protein liquid chromatography, and using a spin desalting column. So cloning, how does this work? So you have your bacterial plasmid, and typically it has a gene for antibiotic resistance. In our case, I know this says ampicillin, but in our case ours, we use kanamycin. And then you also have a promoter region or regulatory region, and we have the lac operon and a restriction site. And so you can use restriction enzymes and cut open your plasmid and insert a gene of interest. And then, so for ours, we had a PET24B plasmid containing our B7H3 Affibody sequence, and also a C-terminal 6' his tag sequence. And what this means that we had 6 histidines linked together, and this is valuable in our purification process. We also had resistance to kanamycin, like I said, and that lac operon. So next step is expression. And first of all, we transform the plasmid into the E. coli, and that just means the E. coli can take up our plasmid, and then we grow that bacteria on a plate supplemented with kanamycin. So that way only the E. coli that has taken up our plasmid should survive. So next, we choose a couple of these colonies to incubate in a lysogeny broth. And a lysogeny broth is a super nutrient-rich media that we can grow a ton of E. coli then. And then we incubate until an optimal density of 0.4, 0.2, and that just means we shine a light through the solution, and once we get to the right optimal density, then we have a good amount of bacteria to work with. And then we induce transcription using I5NG, and that just it looks for that lac operon promoter and allows our E. coli to produce our Affibody in large quantities. So now we have E. coli with our Affibody in large quantities inside of them. So we need to get rid of the cellular membrane. And so first of all, we did this by cell lysis in a lysis buffer, and we hit it with these really high-powered sound waves, and that breaks it open. And so now we're left with a solution of broken up cellular debris. So how do we separate this? We use a centrifugation technique, and luckily our Affibody is insoluble, so we can keep the supernatant. Then we run that supernatant through a column using FPLC, and our column had high affinity to histidine, so we could trap our Affibody within the column and then elute it later. But we're still left with some fragmented proteins. So we used a spin desalting column with the molecular weight cutoff of 3 kilodaltons. So that way we only maintain our Affibody of 7 kilodaltons in size. Lastly, we need to assess the quality of our purified solution, and we did this using a UV-Vis technique called nanodrop. So, after we've successfully created a lot of Affibody, the next step in creating a contrasted agent is binding a fluorophore to it. And so we used, as I mentioned earlier, we decided to use ICG dye. And part of the reason we did this is because it's clinically translatable and already safe to use in humans. So what's the chemistry look like behind this? So we start out with our indocyanine green dye with an NHS ester attached to it. And our Affibody was engineered to have a lot of lysine residues. So that way, this amine group could attack the carbonyl group, and we're left with a stable conjugate as well as an NHS leaving group. So this was a pretty simple reaction. We just combined the two in a tube with sodium bicarbonate buffer. And we allowed it to react for 2 hours, shaking, and we covered it with tin foil so it wasn't exposed to any light. And that's a pretty simplified reaction, but it's good. Okay, so now that we've produced large quantities of our Affibody and we've conjugated it to a fluorophore, we need to characterize this. And we did this using nanodrop UV vis, Sds Page, and a mass spectrometry technique called multi-TOF. So nanodrop UV vis, this is what it looks like. And your sample goes on this little dot in the middle of the black here. And it only requires around 1 microliter of sample, which is pretty great. So it relies on Beer's law. That's absorbance equals molar absorptivity times path length times concentration. And this is this is what's happening. You have a light source, which is emitting a wavelength, a UV wavelength at one wavelength, and then it goes through your sample, reflects off a mirror, and comes back down and hits a detector. In this way, we can measure the absorbance and get a spectrum that looks something like this. So, the peaks I want you to pay attention to here are at 280 nanometers, and this is a protein peak. And so, our Affibody contains amino acids such as phenylalanine and tryptophan, and they have these planar conjugated structures. And then I also want you to pay attention to the ICG dye peaks at 680 and 400 nanometers. And these are just characteristic of ICG here. So our Affibody, without anything conjugated to it, is this blue line. And we'd expect to only see a protein peak. And that's what we see here at 280 nanometers. So that's good. And then we took the absorbance of our ICG NHS, and it should mostly just have its own character, and that's good. And so our conjugate of B7H3 Affibody and the ICG should be a mixture and represent both of these things. And it does a pretty good job of representing ICG, but it's hard to tell with this Affibody peak. So it might just be very dilute ICG. We don't know. So we need to do some more work. And so we did Sds Page. And what this is, it's a technique that allows us to check for sample purity and separate proteins based on charge and size. And so SDS is a detergent which denatures or unfolds the proteins, and it binds to them to give them a negative charge and create uniformity of structure. So now they're not in their folded structure, they're straight, and we can compare them. And this other important part is polyacrylamide gel, and that forms this polymer here. And that'll kind of catch all these heavier proteins so that they take a longer time to move down. And lastly, we apply an electric current, and that's called electrophoresis. So now these negatively charged proteins will move towards this positively charged cathode. And it looks something like this. So we get clear bands based on size and charge. So this is our results. So in setting up our gel, we loaded a ladder in the first lane. And a ladder is a standard solution with proteins of known molecular weights in kilodaltons. And we soaked the gel in Kumasi blue stain, which can bind to proteins, so that way we can visualize these clear bands as they migrate down. In our second lane, we loaded our B7H3 Affibody. And this is a good result because we expect the Affibody to be around 7 kilodaltons in size, and so that lines up appropriately with the ladder. In the next two lanes, we ran our contrast agent. And luckily, and on this right-hand side, we scanned it using fluorescence imaging. And luckily, we see two bands that are slightly heavier than our Affibody peak. So that suggests that we have some conjugation occurring. However, we also realized our purification process following binding the ICG to the Affibody wasn't quite great. It wasn't great yet because we have a lot of free ICG here. But it is still a good result because we have our Affibody and we appear to have some conjugation. So the last technique we used for characterization was called multi-top mass spectrometry. And this stands for matrix-assisted laser desorption ionization time of flight. And basically what happens, you irradiate your sample, you load your sample on a matrix that's on this metal plate, and then you irradiate your sample with short laser pulse of nitrogen gas, which is at a wavelength of 237 nanometers, and this causes our sample to ionize. And then it comes up here through this electrostatic field and separates based on size and hits this ion detector at the top. And this is what your spectrum looks like. So you have mass to charge ratio on the x-axis and percent intensity on the y-axis. And this is a good result as far as we have our Affibody that's been effectively produced because we see a peak here at 7,000. And we expect it to be around 7 kilodaltons. So now that we believe we've created something, we moved on to a biodistribution study, and this took place near the end of my time, so it was kind of a quick run through, and more work still should be done on this. So to start off, biodistribution is a method of tracking where compounds of interest travel throughout the body. So in order to achieve an effective level of contrast agent in the target tissue, the agent must transition from the circulating blood to the tissue of interest and bind to its molecular target. So this study allows us to quantify the localization of contrast agent in different areas of the body. So we did this using a transgenic mouse model, and this means that they were genetically altered to develop breast cancer. And this is good because it provides us with a more realistic genesis of the disease. So we ran the experiment on both tumor-positive mice and transgene negative control mice, and each mouse has around 10 mammary glands, and that's shown here, 5 on either side of their body. So we did a 33 microgram intravenous tail vein injection, which injects it directly in the bloodstream, and we imaged these mice over a 48-hour time period using a fluorescence scanner. And after 48 hours, we removed the organs and imaged them alone, and that's kind of what this is representing, so we could better see accumulation in specific organs. So as I said earlier, these were preliminary studies. So where we were trying to see if this contrast agent even functions as we hoped it would in a biological system, and whether it's actually binding to that B7H3 molecular target, as we had hoped. So this is an example of our fluorescence imaging results. In these images, the strength of signal is denoted by the colored bar on the right, this right here. And so a stronger signal is yellow, whereas a less strong signal is this darker deep red color. We looked at the signal primarily in the liver and bowel because these are the first and last points of clearance from the body, and we also were hoping to see good localization within the mammary tissues. On the mice, this would be a signal here. So the mammary tissues they have 3 under each of their arms and 2 just above either of their legs. And so in these earlier time points, we're primarily seeing signal in the liver and the liver. And then in these later time points, we start to see signal more profuse throughout the body. And on the top, you're seeing the normal mice, and on the bottom, you're seeing these tumor mice. So at these later time points, at 8 hours, 24 hours, and 48 hours, we begin to see a difference in signal strength and localization. So we see a strong signal in the liver here, and not quite as strong, and that might be because it's being retained within the mammary glands there. So lastly, we removed the organs and the mice and imaged them specifically to assess the localization within the mammary tissues and possible other areas of localization. So this image is great to look at and makes some assumptions about results, but we should better quantify this data. So we created some graphs. So in this first plot, we are looking at the average fluorescent signal within the mammary tissue of tumor-positive mice compared to our negative control. The x-axis is the different time points at which we image the mice, and the y-axis is the normalized radiant efficiency, which can be thought of as the amount of fluorescent signal. What this plot reveals to us is that normal mice do not show a significant increase in fluorescent signal in mammary glands. However, the difference between the breast tumor signals compared to normal glands was significant at these later time points, starting at 8 hours. So, when I first saw this, I questioned why do we see an increase in the signal of normal mice at all? And the answer to this is that the signal within the liver was so strong at these time points that it caused this read to be higher. So we had a really high background signal and couldn't get a clear image of the mammary glands. So overall, this is a promising result, and our contrast agent does appear to be binding with the mammary tissue, but it doesn't have as strong a signal as we had hoped for, because it can't overpower that liver signal. So next, in this next plot, the x and y axis remain the same, where the x represents our time points, and the y is our average radiant efficiency. Sorry. So in this plot, we quantify the fluorescent signal within the liver. And the liver is one of the largest organs in the body and plays an important role in removing toxins and waste products from the bloodstream. So the contrast agent we injected localized within the liver early on and may be eliminated or metabolized here. So what we see here is that the normal mice have a peak in liver signal at the 2-hour time point, and the normal mice is denoted by this red line, whereas the tumor-positive mice don't have a peak until the 4-hour time point. So this suggests that the contrast agent is being caught up or slowed down on its way to the liver. So lastly, this plot is complementary to the last plot because it quantifies the fluorescent signal in the bowel. And the bowel is going to be one of the last points within the body where we see signal before it is excreted. Therefore, we don't want to see signal in the bowel too early on in the tumor-positive mice because that would indicate much of the agent was quickly metabolized, excreted from the body, and didn't bind to the breast cancer tissue with high affinity. So in this plot, we see that the agent accumulates to the bowel much more quickly in the normal mice, which this is good because it suggests the Affibody isn't getting held up by anything. In the tumor-positive mice, the signal is much lower and doesn't change as rapidly as in the normal mice. This suggests that the agent is being retained within the mammary glands or other tissues presenting B7H3. So this is all pretty good, but we want even more proof that the contrast agent is being localized within the mammary glands. And so lastly, we did an X-Vivo biodistribution study. So we carefully removed the organs and placed them on a special black background in this configuration, which mirrors what they would look like in the body. So we imaged them, and our result is a bar graph with the different organs on the x-axis and the fluorescent signal on the y-axis. Our sample size is pretty small. We only imaged one tumor mouse and one normal mouse, each with 10 mammary glands. This primary focus was to see if there was a signal within the mammary tissue of the tumor-positive mice and no fluorescent signal within the normal mice. Luckily, this is the case. The tumor-positive mice have a fluorescent signal, whereas the normal mice showed only background level fluorescence. So overall, this contrast agent seems to work in the animal model, which is good. So in summary, breast cancer is a leading cause of cancer-related mortality in women, but if the disease can be caught in its early stages, survivability is significantly improved. Current imaging techniques are insufficient for women with dense breasts, and that's approximately 50% of the population. Current research is focused on developing these photoacoustic and fluorescence molecular imaging techniques so that they may be moved to the clinic, and we created a clinically translatable Affibody fluorescent dye contrast agent that can differentiate between diseased and healthy mammary tissue by both photoacoustics and fluorescence molecular imaging. So some potential future research might include creating an Affibody which binds breast cancer cells with even greater affinity, and also maybe trying out other dyes besides ICG. And also, we had a binding ratio that was pretty small, so we could only bind around 1 ICG per Affibody, and I forgot to mention that earlier. But if you can improve that somehow, that could be pretty effective too. So I'd like to acknowledge my mentors at Stanford, Katie Wilson, Rakesh, and Lotfi, and my mentors at Lawrence, who helped put together this presentation with me, Deanna, Kim, Allison, David, and Daniel. I have Daniel because lab. And I also want to acknowledge the Canary Crest program at Stanford and the National Health Institute for sponsoring this research. Yeah, thank you. Sarah?
So is this lab planning on actually introducing this into the health field, or are they going to try to make it more, make it better before they do that?
Yeah, I think the first steps are definitely to improve it. I don't think it's where they want it to be yet, to actually move to human studies. And photoacoustics is pretty new in that realm. They just recently developed one of these Vivo lasers, which is what we used, to actually be used on human samples. Next?
So did they try running different concentrations of the glowy bit, I guess? I forget what's called. Yes. Did they try using different concentrations of that, or was it just like the one that they tried?
Yeah, so prior to doing the biodistribution study, so I only focused on this because I was working with 2 other interns and we all kind of took different pieces of it. And so prior to doing this, we also did a dosage study to see what dosage works the best for fluorescent signal and signal to noise ratio and stuff like that.
I guess sorry.
Go ahead.
The follow-up would be how long does that stay in the system once it's bound to the tumor?
Well, it appears it gets close to being completely flushed out around 48 hours, but it's not quite there yet, and we didn't go beyond that, so it's hard to say at this point. Yeah. Deanna?
Do you have any ideas on how we would better purify? Because I wonder if some of that background signal is because you just have a bunch of extra dye.
Yeah, that's definitely a good point. And we could just look into, we were using a particular spin column, which just has its own silicon layer, and we probably should have used one with a different molecular weight cutoff. And so we might look into that. Yeah.
Okay, thank you very much.